also. So, okay, reflexes and reactions. That is what we're doing today. And to do that, we'll use a combination of videos, self-testing, and just some explanation. So first of all, what is the difference between reflexes and reactions? So in our case, reactions are voluntary and reflexes are involuntary. But both types of tests test your neurophysiological health. Neurophysiological health. So you can test for things like spinal cord damage, even diabetes, and various kinds of diseases like stroke, for example, or conditions rather. So they're used to test your neurophysiological health. And we are doing several reflex tests and several reactions tests. For your assignment, which will be a report, in your report, you will cover the patellar reflex and reactions. I'm going to start with reflexes. In the report, you won't be reporting on the other reflexes we do today. You'll just be reporting on the patellar reflex, but you'll need to know the other reflexes for quiz purposes for the future. So you will know, so you should follow how the tests are done. So in other words, what is the procedure? What are you testing? And we'll also discuss what types of conditions can affect these tests. Closed captioning, personal closed captioning. So is that okay for everyone? Are you able to follow in the chat? I can't see you, so I have no idea what's going on, but okay, that's good. So it seems like you can follow me in the chat. Good, I'm very happy about that. So what I need you to have with you is your lab manual. So you should have your lab manual in front of you. And I hope that you've read it. We're investigating neural responses to voluntary functions and reflexes, which are involuntary. Outside stimuli can affect your responses. Also, the length of your nerve tract. Also, the size of the neurons that are involved, the diameter of the neurons involved, and how your receptors are functioning, and how your senses are integrating with your motor responses. So integration, which with reflexes occurs at the spinal cord level, and with reactions will occur in your cerebrum. Yeah, so one thing that Gordon McIntyre wrote this lab manual. Gordon McIntyre, he's another anatomy and physiology instructor at Vancouver Community College. And he did add here, which is very true, that measuring reaction time can involve both the subject and the administrator, because the administrator has some response as well. So we will provide our own measurement of response. So reflexes don't require contribution by the brain. They're quite fast. So they are used in particular in response to protective reactions. So I'll write that down. Reflexes are protective responses and largely for postural adjustments. So we're constantly adjusting our posture, but we're not thinking about it voluntarily, or we're not thinking about it consciously. Yeah, so reflexes are very fast. What we're going to do is show you the procedure. So these videos will be online forever. I don't know about forever, but you'll be able to review them afterwards as well. Let's start with the patellar reflex. So for this, I'm just going to show you, not show you, I'm going to give you the link to show the patellar, there it is, reflex. It's not very long, it's just 1 minute and 33 seconds. Copy that. And no, you know what? I think because I'd like us all to watch it at the same time, for these, I will share my screen for the YouTube. Yeah, I think that's a better idea. Okay, here we go. Sharing. Reflexes and Reactions. I'll share the computer sound. If you have any questions or you can't hear it or something, let me know in the chat, please.
So we will start with the patellar reflex, our first part. Kobe will read the manual. I will demonstrate, and Nafisa is going to be our subject.
Have one member of your group sit on the lab bench with their legs crossed, their eyes closed. Tap the patellar tendon gently with the reflex mallet.
First, locate the spot, and then use the mallet. You can use both ways. Okay, we're getting some reflexes here. Okay.
Now we can just show the Gendrastic maneuver, which is a modification to this process. The subject should hold their arm in front of their chest with the elbow extended out to their side. They should lock their hands together by their flexed finger with one hand above the others.
And we'll repeat the same thing. Okay. And then with the other leg too. Okay.
All right. So that was our first one. Okay, good. So what I would like you to do now is your own. Get your blunt mallet. It's called a mallet when you have the proper equipment, but we don't have that. So I'm going to sit over here. I know my image might be a little bit small there. And cross my leg like that. One thing that hasn't been shown in that video is how to measure. Wait, I need something for this, which is a ruler. So while we're doing this in the lab, what happens is You have a subject, that'll be me. You'll have another, and my eyes will be closed. You have another individual finding the patellar tendon. So to find it, you need to find the patella, the kneecap, and it's about 2 fingers down is the best place. So how do you measure that kick? Can you see that? So we measure it with a ruler. Another person is beside the subject, holds the ruler at 0 at the heel. And then when the person kicks, estimates how far the kick is. So that was just one detail that was left out, how to do the measurements. So you take your ruler, you hold it at your heel, and then somebody measures it. So you really need to have 3 people to do this test. But for you at home right now, just try it out. Just see how it works. Try both legs and see how your reflexes are. I'll give you a couple of minutes to do that.
Miss Morn, I have a question.
Yes.
So, starting from the heel, pointing towards like this?
Sorry?
Starting from the heel?
Yes. Yeah, so that's where you would put the 0 of the ruler. You can't really do it yourself. You do need to have someone to do it for you. That would be quite difficult to do for sure, to do it yourself. But if somebody is there helping you, then they can hold the ruler at your heel and just estimate. You can estimate it in centimeters, you know. Is it 10 centimeters, 20 centimeters, 15 centimeters? You know, it's just an estimate, but that is good enough. So once you've done that, let me know how it went. And then try it with what's known as the Gendrasix maneuver. So you can really only do this if somebody is helping you though, because the Gendrasix maneuver is such that you're holding your hands together. So in replacement of that, I suppose you could say grasp an object and squeeze it. So let's try that. You have an object, you're consciously squeezing it, and then try it and see what happens. If it makes any difference. And so when you're collecting data, it's farther, is it? Interesting. Yeah. So that is a question that you will answer for this particular part of the lab. For each section, the patellar reflex and the reactions that we will look at, we'll start with patellar. You need to form an hypothesis. So write down right now what your hypothesis is. Will the knee jerk be greater? Will your reflex be greater with or without this Gendrastic's maneuver? The hypothesis is an answer to this question. Will the patellar reflex be greater with or without the Gendrastic's maneuver? That's the question. Your hypothesis is worded like this: if, then, because. If the Gendrastic's maneuver is applied, then the reflex will be, and you have to choose greater or smaller. Then you write a qualification. Why would that be? Because say you are temporarily distracted from the procedure or something like that. So I put JM for Gendrastic's maneuver. Now the because part, I just sort of loosely put that together. When you make your hypothesis about this test, you use your own language about what you think the reason might be. Your kick might be larger, it might be smaller. So you have to decide, why is that? Why would it be larger? Why would it be smaller? So the hypothesis is an important part of your report. What we're doing today is a scientific experiment. And we're going to be writing a scientific report about that experiment today. So that's your first hypothesis. Now we're not going to do hypotheses for any of the other reflexes today, just this one. And then we'll be doing hypotheses for the reactions a bit later. So let me show you a nice diagram here. So this is an example of a reflex arc. You'll find this in chapter 13 on the spinal cord. There's good diagrams in there as well. I like this one because it's very straightforward. So it shows, for example, the quadricep muscle, which is here, the quadriceps muscle, which is here. And these are the sensory neurons. It's unipolar. So there is the cell body in the dorsal root ganglion, and it carries on to the spinal cord. It ends up in the dorsal root ganglion. So this end here of the sensory neuron is the receptor end. That's known as a muscle spindle. And I'll show you that in more detail in a moment. So that senses this reflex. So you've tapped here, and that has caused your sensors, your muscle spindles in the quadriceps to detect. They've detected a slight stretch. So the reaction is to flex the quadriceps muscle. So this is the sensory neuron, doop, 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 doop. It's going to the dorsal root ganglion, no synapses there at all, it's all the same neuron. But then it goes into the spinal cord, and here it synapses with a motor neuron that goes to the quadriceps, and here it synapses between with an interneuron to the hamstring muscle. Because every time one muscle is flexed, another muscle is extended. So it's flexion and extension. Flexion of the quadriceps, extension of the hamstring muscle. So I'm not going to write all that down because those are the kinds of things you will be talking about when you write your report. But a picture like this would be terrific to add into your report. A picture tells 1,000 words. And I'll show you how to do that later on, how to include it in your report. So another diagram I wanted to show you is this, the muscle spindle itself. So here is a muscle. Here is a tendon with what's known as a Golgi tendon organ. This is the muscle here. Here are the sensory neurons. And the end of the neuron, the receptor part of the neuron, which is kind of like a dendrite, wraps itself around your muscle spindles. And that's how it detects whether your muscle is stretching or flexing. And then that signal goes to the central nervous system or to the spinal cord of the central nervous system. So that's around a muscle cell, and these are the same kind of sensory neuron here. Whoops. But it's interspersed within the collagen fibers of the tendon. And those signals are going to the spinal cord, and then your body will respond accordingly. Okay, so I'll write down here. In your report, you should include some diagrams. Each diagram should have a caption and citation. Yeah, so those are good diagrams to use. Okay, now let's look at the Achilles reflex. Does everyone know where their Achilles tendon is? Achilles tendon. So that is here on the back of your ankle. That's your Achilles tendon. So let's look at that.
Now we're doing the part B, Achilles Reflex.
Have one group member kneel on a chair or a lap bench with their feet hanging free. Have a group member tap the Achilles tendon.
So first I'm gonna locate it, the Achilles, and then tap it. And just observe the foot movement. Okay.
Repeat the process, but this time have the subject grasp an object firmly before the tendon is tapped. Repeat the process again, but this time have them clench their teeth before the tendon is tapped.
So that is the Achilles test, the Achilles test, the Achilles reflex. So the way it's actually done in a neurological setting, like a neurologist, I should say. I think one thing that they forgot, or maybe in the directions, it says that you should bend your foot upwards. So when you're doing this in an office, so your leg is like that, and somebody actually holds your leg, your foot rather, slightly dorsiflexed. Dorsiflexed. So dorsiflexion, if I can do this without falling over. Dorsiflexion is when you flex your foot up like that. That's dorsiflexion. So when you tap the Achilles, what should happen is plantar flexion, down like that. So that's the Achilles reflex. It involves your gastrocnemic muscles, your soleus muscles. So what should happen is a plantar reflex. With an Achilles test, what should occur is a plantar flexion. Yeah, normally the foot is held in dorsiflexion. Yeah, so in this case, we're testing some of the nerves that come from S2, let's see, L5 to S2. So those are the nerves that are being tested with the Achilles reflex. Yeah, okay, let's see. Let's do the next one. The plantar reflex. No, that's not it, sorry. Plantar. There we go.
This is part C, plantar reflex.
Use a blunt probe to firmly stroke the inner side of the sole of the foot. The normal plantar reflex is for the toes to curl down. If the toes flex out and upward, it is called Babinski's reflex.
Perfect.
So there we go. That's the plantar test, or sometimes known as the Babinski test. So before the ages of about 2, the Babinski reflex is dorsiflexion and the toes fan out. But that should only happen before you're about 2 years old. After that, the normal reflex is a plantar flexion. So let me write that down. You can try it. You can try the Babinski. So this is where you use your blunt pen. You can probably just cross your leg over like that and try it on yourself, but you have to do it quite quickly. So they did it quite slowly it's hard to do on yourself. You might not get a response. I didn't get one at all. It's actually probably a good thing. So that is Babinski. So let me write that down. The plantar or Babinski reflex. Normal reflex for an adult is plantar flexion. Before 2 years old, while the nervous system is still developing, the reflex is dorsiflexion, so upwards, and toe fanning. So if the reflex is dorsiflexion in an adult, it could indicate corticospinal injury. So that's what we're testing for with reflex. We're always testing for some kind of injury or some kind of a neurological possible disorder or injury or defect of some kind. So the ciliospinal reflex. Just because it's a bit faster, I think I'll just show you this myself. So the ciliospinal reflex is where you pinch your neck right here. That's your sternomasto, I can never remember. Here, anyway, it's a large tendon right there. And you actually need to pinch it quite hard, so it's better if somebody else does it. And then you watch the pupils. You watch the pupils, okay? So, thank you. Sternocleidomastoid, good. Yeah, so you pinch there. I'm gonna see if I can do this. I'm gonna put, this is gonna be quite funny, but I'm gonna put my eye right up to the camera and see if this works. It's gonna look really funny. Okay, here we go. You ready? Okay, let's see what happens to my pupil. It's already quite dark in here, but did it dilate or constrict or either? It was too fast. Okay. Okay, hang on. Okay. Ouch. I kind of hurt myself there. Okay, so I'm looking right in the camera. Okay, camera. Okay, here we go. I'm gonna pinch now. I can't really tell. No, it'd be hard to see. It'd be hard to see in the camera. So what do you think will happen? So let me tell you this. The ciliospinal reflex, you can try this on yourself. Yeah, you could just hold a mirror right up to your eye and watch your pupil. Let me if you'd be able to do that. Give that a try. See if it works. The ciliospinal reflex tests the sympathetic nervous system. Test the sympathetic nervous system. So that is also known as the fight or flight response. So with the fight or flight response, what would you expect your pupils to do? Remember, you might be in danger, you might be being chased by something. You need to be very alert, but also, Simran, yeah, good. So dilate. You would expect dilation because you can see better. More light will enter your eye and you will have a clearer vision if your pupils are dilated. Yeah, exactly right. So that was the ciliospinal. Nystagmus. Well, nystagmus is a weird one. You can do this at home, but only in a very safe way. So you should sit on a chair that's got wheels on it, or it doesn't even have to have wheels, but the chair itself has to be able to rotate. It doesn't really have to have wheels. Probably better if it doesn't have wheels, but I don't think there are many chairs that rotate that don't have wheels. You can sit on there. Your feet must not touch the ground. You could hang on to the sides. Close your eyes and you bend your head forward by 45 degrees. And then somebody spins you 10 times only. Not too fast. You can try it at home. I don't know if it'll work if I do it myself. Okay, but I'll try it. Okay. I have to push myself with my feet, so it's kind of funny. Okay, here we go. This might take a long time. Okay, here we go. Oh boy. Oh, I'm starting to get dizzy. I can't do it for any longer than that. I gotta put my eyes up. Okay, is anything happening to my eye? It may or may not be. I couldn't quite do it 10 times. It was too much. I don't want to fall off the chair. Oh my goodness. Okay, so that is called, it's a test to see what happens to your eyes. It is fun. Anyway, you can try it. Tell me if anybody actually does it. Tell me, get the person who's spinning you to look into your eye immediately and see what's happening with your eyes. See what's happening with your eyes, if anything. Did anybody get a reaction?
I tried doing it by myself, but I just ended up feeling sick like you did.
Yeah, I know. It's hard to do. Well, I'm going to show the one from the lab. Okay, I'll share this screen.
Can we do the spinning?
Nystagmus.
Rotate Claudia on a rotating chair rapidly 10 times in a clockwise direction with her head tilted forward 30 degrees. Bring her to a rapid stop and
I can't
observe
hear what
the
she's saying.
movement
It's very
of her
quiet.
eyes, direction, nature, and duration.
start, rotate her for 10 times. 1, 2, 3, 4.
I think they'll do a close-up of her eyes.
6, 7, 8, 9, 10.
So we have one more volunteer for this part. Kobe, ready? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
There we go. That showed it. Excellent. Did you see what was happening with her eyes? What was happening? Did you notice her eyes? What was happening with her eyes? Moving side to side. Yeah, they're moving side to side very quickly, and that's called nystagmus. Nystagmus. Yeah. So that's because, you know, she was doing something very unusual her body isn't used to. So her eyes were trying to adjust and find a way to regain her balance, essentially. But oddly, nystagmus happens to people even when they're not spinning. It's just something that happens. So nystagmus is an eye condition. It can be congenital, so it can be present at birth. It can also be acquired, so it can be a condition due to cataracts, for example, can cause it. What else? Eye misalignment also. So, but people with nystagmus generally don't see things as being shaky because their brain just simply adjusts to that. But their vision might not be as good, so they'll probably have impaired visual acuity. So one thing I missed, was the pupillary reflex. So you will definitely need somebody else for this. It's not quite dark enough in here for me to do it, but somebody shines a light into your eye, but you are looking forward, but the light isn't directly into your eye. So make sure you do it at a slight angle and see what happens to the pupil. And then put a piece of paper in between, shine the light in one eye and look at the pupil in the other eye. Like that, and look at the pupil in the other eye. Just to see what happens. What would you expect your pupils to do when the light is shone in it? Would they dilate or constrict? What would happen to your pupils? They would constrict. Yeah, they would constrict to cut out the amount of light. This is too bright. So you can give that a try. And yeah. So if you shine a light in this eye, and this eye constricts, that pupil constricts, why might that be? It's called, interestingly, consensual constriction when the other pupil constricts. So why is that? Think of the destination of neurons. Oh, you can't see. Sorry, that's because I'm still sharing, isn't it? Am I still sharing? Oh, whoops, I sent it to somebody privately by mistake. Hang on. Thank you. Yeah. So the reason is that what happens to the optic nerve? Is the optic nerve ipsilateral or contralateral? Is the optic nerve ipsilateral or contralateral? I've got contralateral, contralateral, and it is also ipsilateral. So interestingly, the optic nerve, some neurons from the optic nerve decussate, some don't, some are ipsilateral. And so because of that, a pupil will respond on one side when when the light is shone on the other side. Yeah, so both occur. It's called hemidecussation. I think we talked about that a little bit before. So what do you think of our at-home lab? Is it working? Excellent. Okay. So now it's almost 11:30, so we're going to take a break. Okay, let's take a 10-minute break, and then we'll get into reactions, which is another part of the write-up, and we'll test some of your reactions. All right, so we'll be back in 10 minutes. Hi, everybody. Are you back? Hi, Tanya. Okay. Good. Okay, so nice to see you again. So we just tested reflexes. So was anybody able to do any of the tests at home? And if so, what is your impression? Tanya, which test did you do? The plantar one. Okay. And it worked out okay? Okay, Amni, you did that one as well? It is quite ticklish. I know. So it's interesting that you should say that. That's interesting because yeah, so you have receptors for tickle that are different than receptors for a deeper touch. So it does need to be quite heavy. Yeah, it does need to be quite heavy. But yeah, even when it's heavy, sometimes you do get the tickle response as well, for sure. Yeah. You were able to test your brother's patience. That's awesome. And your foot went further when you didn't do it yourself. Yeah, that makes sense, doesn't it? Because if you're doing something yourself, you probably would react against the reflex. The cats aren't much good at that kind of thing, are they? Not having opposable thumbs or anything. Okay, excellent. So for the patellar reflex, I hope that you formed a hypothesis and wrote it down. So you'll present that in the introduction of your report. One thing about the tests that we did, the patellar test in particular, is that if you are testing somebody's reflexes, you generally don't do it just once, although a professional could probably just do it once. But since we are collecting data for a report from different people, we do 5 trials and we take the average. So what you would do is you would do it 5 times, the patellar test, and then take the average distance the foot kicked. And that's what I will give you with your data that I will send you. So it's important to do more than one trial if you're comparing different things. In this case, gendrassics and no gendrassics, with and without. So now I'd like to go on to reactions. And to do that, I'm going to show you the video first so that you can see how it's done. So reactions, if you recall, are that, well, let me ask you this, are reactions voluntary or involuntary? What are reactions? Are they voluntary or involuntary? Reactions tend to be voluntary. The way that we're testing them is voluntary. So there are simple reaction tests and there are choice reaction tests. This is a simple reaction test. There's one stimulus and one response in this test. One stimulus and one response only. So I'll show you how the test is done and then we'll talk about it a little bit more. Well, one thing we should do before is this. Because we do need to form our hypotheses. It's probably better to do it before we see the video. So What the subject does is catch a ruler that somebody else drops. It's quite a long ruler. And the thing about the ruler that's cool, that's used in this particular test, is that the measurements are not centimeters, they're in milliseconds. Taking gravity into account, how long does it take for it to drop? That's how long it takes for you to catch it. And it's written right on the ruler, so it's quite cool that way. So we're measuring your reaction in milliseconds. Milliseconds. So that's a thousandth of a second, a millisecond. And we're testing your reaction with your dominant hand and your non-dominant hand. So your hypothesis will answer this question. Which will have a faster reaction? The dominant or non-dominant hand? So that's the question to which you need to form a hypothesis in the if, then, because format. So I'm going to give you a moment to do that. You don't have to write down the because just yet, but just say, if a ruler is dropped, the subject's reaction time will be greater with the dominant hand, or if the ruler is dropped, then the reaction time will be greater with the dominant or non-dominant hand. So that's how you need to write it. Ambidextrous. A person that has cross dominance, or not cross dominance, no, equal dominance is ambidextrous. So yeah, you may be right-handed dominant, you may be left-handed dominant. A left-handed dominance is more rare in the population. The other question is, which cue type will result in a faster reaction? Visual, auditory, or tactile? So once the ruler is dropped and you're about to catch it, would it be faster if you can see the ruler drop? Would it be faster if you've got your eyes closed and somebody says, go, when they drop the ruler? Or would it be faster if your eyes are closed and somebody taps you on the shoulder at the moment they drop the ruler? So you could say visual would be faster than auditory, which would be faster than tactile. You could place them in order of speed. Yeah, and the measurement will be in milliseconds. And you'll see what that looks like when I show you the video. Okay, let me show you that now.
So now we're moving to the second part of our lab, which is reactions. So for this part, we're going to be using a reaction ruler.
Huh.
And I'll just bring it closer to
Okay.
you. So it's a different ruler than usually we're used to. And our measurements, our scale is done in milliseconds. And we start with the thumb
you hear
line,
it?
as you can see it. And we'll demonstrate.
Hold your index finger and thumb approximately 3 centimeters above on the subject. And then have your partner drop a ruler between the two fingers.
Okay, so for the first part of the experiment, Nafisa is going to be looking at me, and I will release the ruler, and then I'll read the measurement. We're going to write it down. We'll have the second part of the experiment, then Nafisa will close her eyes, and I will release the ruler, and the third part, that tactile part, when I will touch her when she's not looking at me. We'll do the same thing. So this is the first part. Okay, it's 150 milliseconds. Kobe will write it down for us. This is for the visual part.
For which hand?
This is a right hand. Okay, we'll repeat the same thing with the left hand. And Nafisa is right-handed. Okay, this is 150 again. This is the auditory part, second. So I will just make a sound or say now, and Nafisa will have her eyes closed. Now.
Well, I don't know why that happened. It stopped in the middle for some reason that I can't fathom.
That was faster.
Oh, start it again. Okay, hang on, let's see if we can finish it then.
Let's see. 110. So we'll choose the right hand. So that's her dominant hand. So this is the last part, the tactile. So I will touch Nafisa on her shoulder when I release the ruler. Okay. So that's 160 milliseconds.
Okay. So that was a millisecond ruler. So if you were designing this experiment when we were at the college, it's important to know a couple of things. Who are your subjects? If we were at the college, our subjects would be college students. So those are your subjects. Another important thing is your sample size. So the sample size of the sheet I will give you is 25. So I'll give you the collected data from a previous class. So that's testing reactions. And reaction tests your neurophysiological condition, your cognitive condition, your information processing. And this particular one was a simple reaction. One stimulus and one reaction. And we tested two different things. We tested handedness, which hand is faster, and also which cue elicits a faster response: visual, auditory, or tactile. So 3 different cues. So now you've formed your hypotheses, which is great. Now I would like to ask you, what kinds of things might affect your reaction times? What types of factors in your life? Would diet have anything to do with it? Your lifestyle, would that have anything to do with it? What could affect your reaction time? What kinds of things affect your neurophysiological condition? You can write that up in the chat. Medications. Yes, medications can affect them, stimulants or depressants. Injuries. Good, that's a good one. Age, sleep. Absolutely. Amount of sleep one gets. Any other ones that might affect it? Caffeine. Good. Diet, such as vitamins. Absolutely. Those are excellent. Yeah, general health, perhaps physical fitness, those kinds of things. Good. So it's those kinds of things that cause variety in your testing population. Those kinds of things cause variety. So what we're looking for is, what is the average of a population? Sorry. What is the average in the population? So with this test, every subject, in this case it would be every student, each one of you, would be tested. And that gives you a good, fairly large sample. And not only that, you would test each time five times. So with your dominant hand, you would catch the ruler 5 times, take the measurement in milliseconds, and get the average. So that's what we've done in previous classes, and I will give you the data. So I'll just show you quickly what that data looks like. Okay, so, and I'm going to send you this, but without the names. So I'll have to cover up the names on the side here or something. So this is what I will send you. So you've got 4 columns. You've got visual cues with your dominant hand, visual with your non-dominant, auditory, and tactile. So what you'll be comparing is these 2 here. And you'll also be comparing these 2 and this one. So you'll be comparing visual with auditory and tactile. So you'll be using this visual column twice. You'll be using that column twice. And you will have to estimate the averages. And there's something else down there you need to estimate, which is the range. So now I'm actually going to give a bit of a lecture about how to write your report. So I do need to do some writing. So I'll transfer over to my iPad so I can write some things for you. So are there any questions about how the tests were done at this point? And you know, those videos will be up there. Now, one thing that is important when you're doing a test like this with a whole classroom of people, you need to administer it in exactly the same way. So if, for example, you're catching the ruler, you should determine in advance whether your forearm should be parallel to the floor, perpendicular to your body. So you should determine that ahead of time so that everybody's doing exactly the same thing. So that's very important when you're conducting an experiment with lots of different people. You need to administer it in the same method, using the same method. And so, yeah, those kinds of points are important. So let me take another break now. We'll all take a break, but this time just for about 5 minutes, okay? And then we will regroup on with a little bit of an introduction to how to write a report. Okay, I'll be here, but I'm going to stop the video and mute, but you can ask me any questions you want in the chat.